Hello, fellow warriors. Here we are with news from the website, prostatewarriors.com.
It's great to be here.
Welcome to this deep dive. You know, if you've been tracking the landscape of prostate cancer over, say, the last decade, you know we've largely been fighting a war of attrition.
Yeah, it really has been a tough road.
Right. I mean, the traditional playbook has been somewhat reactive.
Hmm.
We deploy a systemic therapy, we wait for the tumor to inevitably develop some kind of resistance mechanism, and then we just try to pivot to a new agent.
Which works for a while, but it's fundamentally a defensive posture.
Exactly. But the stack of research we are unpacking today, it points to a massive paradigm shift. We are moving from that defensive posture to a highly predictive precision strike offense.
We really are. It's a critical moment in oncology right now.
Yeah, we're diving into next-generation radioligands, drugs that literally hijack a cancer cell's internal disposal system, and genomic data that might eventually let us predict a tumor's exact evolutionary path on the very day of diagnosis.
We are no longer just trying to broadly suppress the disease. We are looking at exactly how the cancer manipulates its microenvironment, and its own genetic code to survive.
We're getting into the microscopic detail.
Exactly. The clinical trials and the real-world data we're reviewing today reveal how we can dismantle those specific biological defenses. It's dense, complex science, but it points to a future defined by truly personalized, aggressive intervention.
Okay, let's unpack this. Because per our focus on the front lines, we have to start with the latest clinical trials. Specifically, we're looking at the new frontier of targeted radiation.
Right, a huge area of development.
Yeah. I mean, we know that 177Lu PSMA 617, which most of you probably know as Pluvicto, has been just a foundational game changer for metastatic castration-resistant prostate cancer.
Absolutely, it changed the landscape.
But now we're looking at a phase 1 trial for a next-generation approach. It's a new small molecule radiopharmaceutical called 177Lu PSMA VG01. So how does VG01 actually improve upon the PSMA targeting architecture we already have?
Well, to really understand the upgrade, we have to look at the physics of how a lutetium-177 payload actually kills a cell.
Okay, lay it on us.
So it's a beta emitter, right? When the molecule binds to the PSMA protein on the surface of the cancer cell, it releases a high-energy electron.
A beta particle.
Exactly, a beta particle. And that particle travels a very short distance to induce highly lethal double-strand DNA breaks in the tumor. But here is the critical vulnerability with that.
Which is?
That process takes time. If the radioligand detaches from the cell receptor too quickly, if it washes out, the radiation dose is just deposited harmlessly into the surrounding fluid.
Oh, wow. Or worse, into healthy tissue. Right.
Yeah.
Instead of shattering the tumor's DNA.
Precisely. That's the main issue.
Okay, so it's basically a matter of grip strength. Because the preclinical data for VG01 shows a 2 to 3-fold higher binding affinity. So it's like upgrading a targeted missile so it not only locks onto the target better, but also drastically reduces collateral damage to innocent bystanders. In this case, the kidneys and bone marrow.
That is a perfect analogy. The preclinical models demonstrate highly sustained tumor uptake. And because it binds so tenaciously to the tumor, there's less of the active drug just left aimlessly circulating in the bloodstream.
Which improves the tumor-to-kidney ratio, right?
Precisely. Small molecule radioligands are primarily cleared from the body through the kidneys. If they hang around the proximal tubules too long, the kidneys absorb a massive amount of collateral radiation.
And renal toxicity is one of the primary dose-limiting factors in this kind of therapy. Like, if you damage the kidneys, you have to halt the treatment, even if the tumor is still active.
Right. But VG01 accelerates systemic clearance from non-target tissues, so it showed minimal bone marrow activity and a vastly better tumor-to-kidney ratio. Theoretically, this allows oncologists to administer a significantly higher, more curative dose of radiation to the tumor while preserving the patient's renal function.
Maximum lethality for the cancer, minimum collateral damage for the patient, widening that therapeutic window. Now, precision radiation is only one piece of the puzzle. Here's where it gets really interesting. When we look at standard systemic treatments, the tumor's primary survival strategy is mutating its internal machinery to resist our drugs.
Right, overcoming hormonal resistance is a massive hurdle.
Yeah, which brings us to a highly anticipated Phase 1b trial. This is looking at a drug called Bavdegalutamide, or ARV-110, combined with Abiraterone. This trial included 45 patients with metastatic prostate cancer whose disease was actively progressing on Abiraterone.
This trial is fascinating because it tackles one of the most frustrating mechanisms of resistance.
Yeah.
The androgen receptor ligand-binding domain mutations.
Right.
Drugs like Abiraterone exert enormous selective pressure on the tumor. So to survive, the cancer frequently develops mutations.
Mm.
Once the shape of the androgen receptor's binding pocket changes, standard competitive inhibitors just can no longer dock properly. The drug becomes useless.
Standard therapies just block the androgen receptor, but bavdegalutamide is an androgen receptor degrader. Is that the difference between, like, putting a piece of tape over a lock versus completely dismantling the door?
That is the exact analogy I would use, yes. Because you're absolutely right. Standard inhibitors are just trying to jam a key into a lock that the tumor has deliberately whopped. But bavdegalutamide bypasses that entirely. It's a PROTAC.
A PROTAC. Okay, how does that actually work?
Well, it's a two-headed molecule. One end binds to the androgen receptor, and the other end binds to an E3 ubiquitin ligase.
Which is basically the cell's internal tagging system for garbage disposal, right?
You got it. It forces the ligase to tag the androgen receptor with a chain of ubiquitin molecules.
Oh, wow. And then that ubiquitin chain acts as a homing beacon for the 26S proteasome, which is basically the cell's biological incinerator.
Exactly. You are physically eliminating the target from the cell. Because you are destroying the receptor entirely, rather than just trying to block its active site, it remains highly effective, even in patients with those stubborn AR ligand-binding domain mutations.
And the clinical data from this phase 1b trial really underscores how potent that mechanism is. I mean, even in a heavily pretreated population progressing on abiraterone, this drug showed a PSA control rate of 48.9%.
That's a very strong signal.
Yeah, and a PSA 50 response in 24.4% of patients, plus an encouraging median radiographic progression-free survival of 16.3 months.
And we really must emphasize the safety profile here, too. With novel mechanisms like protein degraders, off-target toxicity is a major concern. But the toxicity here was mostly just low-grade fatigue and nausea.
So bavdegalutamide destroys the internal machinery of the cancer cell. But what happens when the tumor builds an external biological shield to keep our therapies out entirely?
Ah, the microenvironment.
Right. That takes us to a phase 1/2 trial evaluating a drug called SX682 in combination with docetaxel in advanced prostate cancer.
This shifts our focus completely. We really have to stop viewing tumors as just isolated clumps of rogue cells. They're complex ecosystems.
Right.
In advanced disease, the tumor actively secretes chemical distress signals called chemokines. These bind to CXCR1 and CXCR2 receptors on immune cells. The tumor uses them to deliberately recruit corrupted immune cells called myeloid-derived suppressor cells, or MDSCs.
And these MDSCs, they're essentially a cellular defense matrix, right?
Yeah.
Like they blunt the body's immune response.
Exactly. They secrete factors that physically suppress and exhaust the patient's cytotoxic T cells. They create this localized immunosuppressive moat around the tumor.
So docetaxel is the standard heavy-hitting chemotherapy, but SX682 strips away the tumor's immune defense so the chemo can hit harder.
If we connect this to the bigger picture, bringing SX682 into a taxane-based chemotherapy backbone solves a massive problem: treatment resistance and immune escape in MCRPC. SX682 is a CXCR12 inhibitor. It blocks those chemokine receptors.
It cuts the communication line.
Exactly. The tumor can secrete distress signals, but the MDSCs never receive the homing signal. When you strip away that immune suppression, you fundamentally alter the efficacy of the chemotherapy.
It's a brilliant synergistic approach.
Mm-hmm.
Now, all of these trials represent the bleeding edge of experimental biology. But moving from experimental trials to the real world, how do doctors choose the best combinations of the therapies already available on the market?
Yeah, real-world data is crucial for that, like the ARON3 study.
Right, the ARON3 study. This was a retrospective comparison across 14 countries. It involved 247 patients with metastatic hormone-sensitive prostate cancer, or MHSPC.
And it compared two major triplet therapies.
Right. It was darolutamide plus docetaxel plus ADT going up against abiraterone plus docetaxel plus ADT. Now, the unadjusted data showed progression-free survival heavily favored darolutamide. Like, the median wasn't even reached for the darolutamide group compared to 21.5 months for abiraterone.
The raw numbers definitely favored it, yes.
So what does this all mean? Is darolutamide the undisputed champion here?
Well, we have to look at this with a calm, nuanced view. Real-world data is messy, right? Doctors don't randomize patients in practice, so researchers apply statistical adjustments to balance the groups.
Not like propensity score matching.
Exactly. And after those statistical adjustments, the differences weren't technically statistically significant anymore. But, and this is important, the trend still favored darolutamide.
And it was especially clear for a specific group of patients, right?
Yes. Darolutamide showed a clear edge for patients facing the most aggressive manifestations of the disease, specifically those with visceral metastases and high-volume disease.
Where every marginal advantage really matters. And what about safety?
Safety was largely comparable across both, though Abiraterone did have slightly higher rates of grade 3 to 4 fatigue.
Okay, so that's optimizing systemic drug therapy. But optimizing care isn't just about drugs, it's also about how aggressively we deploy radiation when the cancer has spread to the bones.
Right, metastasis-directed therapy.
Yeah, so we have this retrospective study of 246 men across 347 radiotherapy courses. They were looking at intensified radiotherapy for bone metastases alongside standard prostate-directed radiation.
And this is a big shift, moving away from purely palliative radiation where you just treat for pain.
Right. And the headline was that higher dose bone radiotherapy was linked to significantly better 5-year survival. But there's a specific subgroup I want to look at. Patients with no visceral metastases treated within the first year. The median overall survival was 40 months with aggressive radiation versus 26 months without. And the 5-year survival was 47% versus 18%.
Huge clinical differences.
Massive. But wait, the source notes the p-value for that specific subgroup was 0.11, meaning it wasn't statistically significant. Should you, the listener, still care about these numbers?
Yes, absolutely. I mean, we must respect statistical significance, but a p-value of 0.11 just means the sample size was small. The sheer direction of the effect perfectly aligns with a larger medical paradigm shift.
Moving away from palliative care.
Exactly. Moving toward aggressive metastasis-directed consolidation treatment for low-burden disease.
But this brings up the ultimate logistical hurdle. We have intense triplet therapies and aggressive radiation. But how do we know which patients actually need them?
That is the big question. We don't want to overtreat a slow-moving tumor or undertreat a vicious one.
Right. And that brings us to a foundational genetic discovery published recently in Nature. They looked at gene expression across normal, primary, and metastatic tissue. The goal wasn't to see who gets cancer, but why some tumors stay quiet while others become life-threatening.
Right. They were hunting for the evolutionary drivers of metastasis.
And they pinpointed a region on chromosome 1. A gene called RCC1 stood out as the strongest candidate. Higher RCC1 activity was unequivocally linked to a shorter time before the cancer returned after treatment. The study also highlighted the PHACTR4 gene.
Yes, RCC1. It basically unhinges the cell cycle so the cancer cells divide relentlessly.
It's like finding the exact line of code that turns a benign software program into a vicious computer virus.
This raises an important question, really. While this isn't definitive proof of cause and effect quite yet, it proves that understanding prostate cancer biology is shifting. We are moving from just diagnosing its existence to predicting its aggression on day one.
Which is incredible.
Yeah.
Well, to quickly recap the journey of this deep dive, we've covered a lot of ground. From the high-tech precision of 177-Lu PSMA, VG01, and Bavdegalutamide, to altering the immune microenvironment with SX682.
We really went from the micro to the macro.
Yeah, we used real-world data to look at optimizing triplet therapies, analyzed aggressive bone radiation, all the way down to the genetic roots of cancer aggression with the RCC1 gene.
It represents a staggering amount of innovation, and I just want to leave the listener with a final provocative thought to mull over.
Good for it.
If we can eventually use genes like RCC1 to predict a highly aggressive cancer on the exact day of diagnosis, could the future of medicine mean skipping standard hormonal therapies altogether?
Oh, wow. Just bypassing them completely.
Right. We might one day go straight to targeted agents like 177-Lu PSMA VG01 or CXCR12 inhibitors before the cancer even has a chance to mount a defense.
Anticipating their move and ending it early. That is an incredible vision of where this is all heading. Well, thank you so much for joining us on this deep dive into the science. Stay strong and fight on.